Our next speaker is Dr. Fabio Falleroni. Dr. Falleroni is a neurobiologist and clinical researcher at IRCCS Ospedale Policlinico San Martino in Genoa, Italy. His research focuses on the cellular and molecular mechanisms underlying neuronal function and dysfunction, with a strong interdisciplinary approach integrating neurobiology and biophysics. He earned his PhD in neurobiology from the Scuola Internazionale Superiore di Studi Avanzati and completed postdoctoral training at the Technical University of Munich. Dr. Falleroni is known for developing oscillatory optical tweezers, an innovative technique that enables the application of piconewton scale forces to study cellular mechanobiology. His current research explores the mechanism of neurodegeneration across the lifespan. Dr. Falleroni has also been engaged in studying UGDH as an awardee, I believe, of UGDH Italia's last grant cycle. So now let's hear from Dr. Falleroni.
Thank you so much, Rebecca, for the nice introduction, to give me the opportunity to share my project, to be part of this special meeting of your special family. So the title of my presentation is Development of a Conditional Mouse Model for Jamuar Syndrome. It seems so there is a huge spot in Italy that we are trying to create a mouse model for this disease. And actually, I got the model. So thanks to the telethon, as you said, and the UGDH Foundation, the Italian Foundation, we generated the first Jamuar syndrome animal model by creating a knockout mouse. So basically reproducing the loss of function phenotype characteristic of the disease. In order to do that, we use the Cre-lock system. We saw this system in the previous presentation. And basically, the Cre-lock system is composed by two components. One is the Cre enzyme, as you can see the red box, and then you have the LOXP site that actually here are at the both ends of the UGDH gene. So let's see how the Cre system works. So the Cre system derived from a virus, actually in a specific, the virus P1. It's a particular virus that mostly infects bacteria. So when it's inside the cell, the virus, it has a linear DNA, double-stranded with the LOXP site. So when it's inside the cell, the bacteria, the virus start to use the Cre enzyme to cut the sequence, the DNA sequence between the LOXP site and make it circular. So in this way, the bacteria, when the viral DNA is circular, the bacteria start to replicate the viral DNA. So the virus took advantage of this system for ages, and now we are using the same system in the laboratory to perform some genetic manipulation. So we adapted the system, and now we are able, like we saw before, to insert the LOXP site at the bottom end of a specific gene, and then we use the Cre enzyme to cut the sequence to obtain a knockout chromosome, allele. So as we saw before, the huge problem with this disease was to create an animal model, because the full knockout, so when you delete completely the gene from the body, it's completely lethal. So we to overcome this, we use a nesting promoter. So we created the Cre-lock system, and then we bind the Cre-lock system with the nesting as promoters. So if you ask me what is a promoter, it's a molecular switch. Basically, the cell, when see the promoter, start to express the gene that is close to the promoter. So by doing this, since the nestin is expressed just in the nervous system, starting from the neuron progenitor cell, we can remove the gene just in the nervous system, a neural compartment. As you can see here on the bottom right, there is the expression of the nestin in blue. As you can see, the expression track just the central nervous system. So I generated the animal. So I started this project in January when I received the grant from the Teleton Foundation. And basically, I spent all the months, 5 months, just to generate the animal. So now we have the pups in which the gene is completely shut down just in the nervous system. The pups are alive, are quite, they behave normally and are physiologically. It's a physiological model. So the first phase of my project is about ex vivo experiment. So basically ex vivo means that we take neuron from the brain, from the pups, in which we deleted the gene, also from pups in which, what type pups in which the gene is completely functional. And now I'm going to perform, I'm starting to perform experiment using this special device that are called multi-electrode arrays. Basically, are the hardware composed by several electrodes embedded on the surface. Those devices are transparent. You can place the neuron there. And by using the electrodes, you can record the electrical activity of the neuron. Because yes, the neuron develop electrical currents in order to communicate each other. Those are very preliminary experiments because I started to perform this kind of experiment 3 weeks ago. But are preliminary, but you can see a huge difference between the wild type and the neuron from the wild type and the neuron from the UGDH knockout. These are very preliminary, but there is a clear over-excitability on the knockout neuron. Now I perform also standard experimental procedure like histochemical analysis. Here, basically, we can track, we can follow a specific compartment, a specific protein inside the neuron, how they develop. For instance, here we have the nuclei of the neuron. In green, the actin that composes the cytoskeleton, that basically is the structure, the physical structure of the neuron. And then at the bottom, we have we stain two different plasma membrane channels that basically play a key role in the maturation of the neuron during the development. And then on the right, we have the merge of the four channel. So what I'm saying that we just generated the animal and then we are performing all the experiment that basically are divided into phase. The first phase is the ex vivo phase in which we take the neuron and we grow them in the lab in a dish and we observe at the cellular level. And then we will go on in the second phase by performing in vivo experiment. So basically we can track how the mutation affect the neural development after the birth. So after birth. So this is a very simplified presentation. It was my first time here at the UGDH Congress. So basically that's all from my side, but for sure, if you are interested, just contact me or ask me and we'll do, for sure we'll do a discussion. My field, as Rebecca said, is about the mechanobiology. So I will focus my attention at the interface between extracellular matrix and neural development, both at the pre-birth and after birth of the animal. So for now, that's all from my side. Thank you for your attention. Thank you to all. Yeah.
Wonderful. Thank you both, Dr. Colasante and Dr. Falleroni. And as Rebecca mentioned, we'll take questions for both in this session where it looks like we've gone from 0 mouse models to 2 mouse models. So that's always a fantastic thing. The more models, the better. Please put your questions in the main chat, in the Q&A, or raise your hand and unmute. I'll start with, so we have a conditional knockout where we can look at the effect of loss of UGDH in the CNS, and we have a missense variant mouse model. Could you both speak to the different ways that both of those models could be used?
Yeah, I mean, I think that it's a complementary approach, but I will leave Gaia to speak.
Yeah, I mean, yeah.
Yeah.
Yeah, generally, I mean, in particular because genetic of mice is a little bit different from humans, so we don't really know how much the mouse model will reproduce the phenotype of patients, and in particular, which dosage of the protein will be necessary in the mice to have symptoms. So I think having both approaches is extremely helpful. So the knockout does not have the protein completely, so there is no UGDH protein into the brain, while in our case we will have a double, I mean, we have both allele encoding for a mutant protein. So the protein is there, but easy to function. And I'm really, I mean, I take advantage of this Q&A moment to also, I mean, acknowledge Dr. Melanie Simpson for, I mean, explaining and characterizing very well the mutation. And I have a question actually for her. So if you think that we should change, I mean, we should have a different diet for our mice in order to have a more clear phenotype. So we should have like a low glucose diet or something. I don't know.
Yeah, that's a great question. What I would say about that actually is that the availability of glucose fluctuates naturally during development, irrespective of the diet of the mother.
Okay.
And so I would say that for the developmental model, having the diet be as similar as possible to what the diet would be is the best. Why we manipulated glucose levels in our studies is because especially when you have an ex vivo type of a culture system, it's really very dependent on the culture system that you set up. And so we were trying to simulate the types of situations that would happen naturally in vivo, but I don't think that you need to focus on that specifically. It may be that some like caloric restriction studies might be interesting because I could see that being important for seizure management even, but I wouldn't be excessively worried about that for the development of the mice.
Thank you so much.
Beautiful study.
Yeah, thank you.
Yeah, regarding the model, the nesting model, yeah, for sure the gene is completely deleted, but what I got that in some case the activity of the enzyme is so low that you can detect the activity. So basically, in some cases, I think that the enzyme is not working at all. So we are shutting down the gene just in the neuron. So just in the nervous system, just the neuron cannot produce the extracellular matrix, but there are other components that can produce extracellular matrix. So it's not full physiological, but I think that replace the disease very well. I mean, if the measures the symptom of the UGDH disease is the developing of the epileptic seizure, I mean, by using this model, we can see that they have several excitability. So we are reproducing exactly the phenotype. It's a mouse model. There is a huge overlap between us and the mice, but for sure, it's the most relevant model that we can have until now.